Good day, everybody. My name is Michael Ray. I am science writer at SENS Research Foundation, and I'm joined today by Dr. Sudhir Paul. Dr. Paul is Chief Scientific Officer of Covalent Biosciences. That is a rejuvenation biotechnology working on antibody-based therapies against a variety of targets, a lot of which are really relevant to aging, and includes a platform technology that we're going to be discussing in some detail. Dr. Paul, good morning.
Good morning. Thank you for having me here.
Thank you. So what's the big problem you're trying to solve?
So we are focusing on aging. We, as we know, living life in general inevitably results in accumulation of toxic byproducts that can be collectively called detritus that must be gotten rid of. And an example is amyloid proteins that are formed within minutes to seconds in a test tube, but in the body they are formed more slowly because there are processes that retard the amyloid formation. By the time we are in our, let's say, the 20s or so, you can already detect increasing formation of these aggregates of proteins called amyloids that eventually become particulate. Initially, they are soluble and oligomeric. Subsequently, they deposit in the tissues and start causing big problems, diseases, essentially.
And we should just be clear that you're using amyloid in the very general sense. A lot of people will have heard of beta amyloid, which is one particular kind of amyloid that forms in the brain. But there's a lot of other proteins that form these same aggregates you've been describing and that affect different tissues because of the way that, the tissue where the protein appears and the particular metabolic conditions that are in a particular tissue and that kind of thing.
Absolutely. So you are so right about it. That's important to point out. The amyloid formation, these proteins are normally essential proteins when they are not misfolded and then aggregated. Folding is the three-dimensional shape of the protein. There is a motif called the beta sheet motif. So this is found in all proteins essentially. And these proteins have somewhat more of that shape motif that results in their aggregation. So the misfolding process, normal folding, involves only a small amount of the formation of these aggregates that's well kept under control by our physiological living processes, and we remain young until, as we were discussing, when we are young, these proteins do not accumulate too much, but with time, as we get older, they form increasing amounts of particulate residues and deposits and so forth, and disease occurs, both because of anatomic disruption of various organs, the brain or the heart or the lungs and so forth, and because those soluble oligomers of these aggregated proteins are toxic for cells. So there's a direct toxicity, and also the fact that you are disrupting the anatomic architecture with increased amyloid deposition results in major diseases. We are focusing on three different amyloids that accelerate aging. One is transthyretin, a protein that is important in heart disease in older people, but even at a younger age already, musculoskeletal problems like carpal tunnel syndrome and spinal diseases, which are really caused by the transthyretin amyloid protein deposition, they start appearing a little bit in mid-age, midlife, and then as we age, the diseases show up in a life-threatening manner. That's the systemic amyloid diseases caused by transthyretin amyloid. And then, of course, we are also focusing on the more well-known beta amyloid that you mentioned at the outset, along with tau amyloid, which has been more recently highlighted as a contributor in Alzheimer's disease. There is the amyloid hypothesis of Alzheimer's disease, which is certainly partly true, but various features of that are being refined through fundamental science. So a lot goes wrong in Alzheimer's disease because the process of amyloid formations appears to initiate the disease, but then inflammatory processes come in a big way, and the brain eventually loses its capacity to execute its various functions, thinking and memory and decision making and so forth. So those two targets, the A beta and the tau in our studies is a little less advanced, but for amyloid beta and for transthyretin, we have lead antibodies that are ready to go to the next stage of clinical testing. Now,
Right.
for
Now, you're
amyloid,
developing in particular two kinds of antibodies, one of which is a really exciting platform called Catabodies. Explain to us what Catabodies are and how they differ from better known binding antibodies.
right? So we have a toolbox. We produce catalytic antibodies and irreversible antibodies out of certain discoveries, basic science discoveries I made with my group at the University of Texas. I've been in this area on antibodies. I've been working on antibodies for about 40 years. We discovered that some antibodies have chemical reactivities. So they go beyond what a conventional antibody does, which is simply non-covalent weak binding. That's what a normal antibody does. It binds and then it stays bound for some time. That binding is reversible, and because the binding is weak, there is dissociation of the immune complexes. We discovered what are called in chemistry nucleophilic antibodies, which have, as the name implies, they have a love, they are philic for positively charged nuclei, which is the basis of their covalent, very strong reaction with electrophilic substances in, so these are proteins are electrophilic, they're weakly electrophilic, and there is a covalent reaction of these antibodies, nucleophilic antibodies. If the antibody, we call it an active site, the chemically reactive site is the active site. After the covalent bond is formed, if there is support for water attack on the covalent complex, we get breakage of peptide bonds. And after the peptide bond breakage has occurred, the antibody is recycled. So it's a catalytic molecule because it is recycled. That's a part of the definition of the word catalytic, that it's reused again and again for many reaction cycles, which results in an increase in the efficacy because we are now taking a small amount of a catalyst and mixing it with a large amount of the target. And within minutes to hours, we can get complete degradation, like amyloid degradation is the specific example that I'm citing here. A lot of amyloid.
I'm just gonna put a point on that, Dr. Paul. So the key point I think for our listeners here is that a conventional antibody, it binds with more or less affinity to its target, and its mechanism of action as a therapeutic is usually just that it's neutralizing its activity, but you can only get one antibody to one target molecule, whereas the catabodies, these catalytic antibodies, when they bind, they have this additional feature that they will actually cleave bonds inside the target and eliminate it altogether. And that allows them to release and go on to the next molecule and cleave, and the next molecule and cleave, and so forth in a catalytic action, which allows one antibody to completely neutralize many, many molecules of the target. Is that a fair summary of what you've been saying so far?
That is well said, and better than I could have said it. I'll just say the word neutralization. There's a lot in semantics and terminology that we have to be careful about. And the word neutralization originated classically from just the binding, that you have an antibody that binds the target. That portion of the target is sterically blocked. So it's hidden, it becomes hidden due to the binding. When this comes off, when it dissociates, this becomes activated again. An irreversible bond, it just stays bound. A catalytic reaction means my left hand is going to be broken into its knuckles and into its component parts, into inactive parts. I won't have a hand anymore. And that is what should happen to detritus like amyloid, which is not.
That's a great metaphor, and I'll actually just extend that if I may. So the idea here is a normal antibody is going to bind that hand and prevent it from gripping onto something. Here, you're actually cutting off its fingers, so it can't grip onto something. Yeah, that's excellent.
It becomes incapable. I should say, Michael, that we discovered these antibodies as naturally occurring antibodies. I had made a whole lot of song and dance earlier about early in youth, we have accumulation of the amyloids, but it's a slow process. In the test tube, this happens within seconds to minutes. My point is, these antibodies that we discovered in humans and in other mammals, they occur naturally. They are a defensive mechanism for retarding amyloid accumulation. Normally, things are kept in check. Normally, that is, normal means youth. As we get older, things become abnormal, and the equilibrium, the removal of the amyloid is no longer balanced with the production of the amyloid, with the result that there is accumulation of the toxic amyloid. So we think that when the amyloid is produced in very large amounts, the amount of the antibodies is not sufficient to control the disease, to amyloid disease. We would like to now get our recombinant catalytic antibodies that we isolate using a technology that we call electrophilic protein analog technology. We pull out the best catalysts and the best irreversible binders from these libraries. And then we want to supplement the natural defense that humans have against amyloid disease by either intravenously injecting, which is the common way that antibody therapy is done, that they are injected intravenously. That's the major option that is currently available. We've also shown by gene therapy, when we take the gene of in fact, the Alzheimer, as we call it, this is the amyloid beta directed catabody, and we put it into an animal model of Alzheimer. So we can deliver the gene for the catabody, and we can remove amyloid from the mouse model, the brains of the mouse model, through the genetic means rather than through intravenous injections. Both options are open. A gene therapy is a less validated technology currently. The intravenous delivery technology is the common way of doing therapy with antibodies.
So you've looked at these, as you've said, for several targets now. The one that was originally sponsored by SENS Research Foundation was for transthyretin amyloid, which again was the one that causes age-related heart failure, amongst other diseases. and also for A beta and tau amyloids, which are involved in age-related cognitive decline and dementia. Are there any targets for which catabodies are a bad choice?
Yes, so generally the catabodies degrade the protein. Now, if the fragment or the product fragments that we observe due to the catalytic reaction, when the target is broken, it breaks into smaller peptides and polypeptides. If the polypeptide, the product polypeptide, is more active than the precursor, that is not desirable if our goal is to remove the toxic species. And that we've in fact published, others have published that in some cases we have identified catalytic antibodies that can activate. For instance, in our own case, in our publication, we showed that prothrombin is broken to form thrombin. And thrombin is an enzyme that is used in coagulation, blood coagulation. So we don't want that kind of thing happening with our amyloids. And the reaction is variable from target to target. In some cases, it is an activation reaction. I would say, in the minority of cases, if we want, for instance, a coagulation factor treatment for heart disease, we would be looking for a thrombin. So in cardiovascular disease there are blood coagulation issues there. If we want to break up the blood clots, we might be looking for such an antibody, but not for amyloids. So that's an example that we would reject such a catalytic antibody. So we have to be careful. The whole concept of selection of the antibodies is based on the specificity of the antibody for the target, meaning it should react only with the target. There should be no off-target effects. Then further specificity issues for us in the catabody field are that the digestion, the breakage of the target should not result in an active fragment as opposed to an inactive fragment, at least for the amyloids. And the good news is that we have managed to apply our selection algorithm in a way that we ended up with lead products, lead catabodies for TTR and for A beta, the TTR being transthyretin, A beta being amyloid beta, that produce non-aggregogenic, so that the product fragments are not aggregogenic, whereas the starting
meaning not prone to aggregate.
Exactly, it doesn't form the aggregates, and the product fragments are also non-toxic as compared to the starting material, the amyloid aggregated proteins that are toxic.
Great. So those are your leads. They all have all the properties you would want in terms of successfully breaking down the target and not creating any nasty off-target effects. How are things going? What's your progress been?
Our progress is good, but slower than I would like, which I guess there are multiple determinants of our progress. We have first the focus on the cardesign that I mentioned.
This is again the TTR binding or TTR degrading catabody.
So that lead product, we know its sequence, we determined the sequence, we've done specificity analysis, and this antibody is really unique, that it is exquisitely specific for only the misfolded form of the TTR. The normal TTR protein is not recognized by the antibody. It's not broken by the antibody. So which is good news, because we will not interfere with the natural functions of the transthyretin protein. These proteins are, as I said earlier, they do have important functions when they are not misfolded. So we don't break the misfolded TTR at all. We have also shown that there is potent and rapid removal of TTR ex vivo, so outside, not in vivo, but tissue sections. When they were treated with the cardizyme, we got very rapid and potent, within minutes to hours, we were able to remove all of the deposits. Then our second focus is not going so badly either. We have in vivo mouse studies with a stabilized form of we call it Alzheimer, in which we incorporated a tag called the FC tag. Now, these all technical things are milestones for us. They might not be easily understandable at face value, but how is it going? The going is defined by the achievement of these technical milestones that I'm mentioning, that we have stabilized the protein and it now can act for about 20-fold longer time than our original Alzheimer. And again, this one also this one clears not only ex vivo, but also in vivo in mouse models. I already mentioned that we've shown both by intravenous injection as well as by gene therapy, clearance of brain amyloid A beta. We also
The
have
tagging process you were mentioning, the idea there is that if you can get the protein to hang around longer in the brain, then once you've gotten into the brain, it has a longer opportunity to cleave more aggregate molecules, and therefore each molecule of the drug, where the Catabody is more potent in terms of how many molecules it can degrade and how rapidly it gets all that dealt with.
Right. So essentially, we are into the translational component of the project. In other words, we have a lead product, and now we are optimizing it. And that optimized product, we are close to signing a contract with a major company with late stage experience in drug development. Remember, I was a basic scientist. Now I'm more of a translational scientist, but I need help from colleagues and other companies with this kind of experience. We call it pharmacokinetics, improving the pharmacokinetics in the brain. In this case, there's also the uptake issue, that there's a blood-brain barrier. We showed that our Alzheimer can cross the blood-brain barrier successfully and accumulate at respectable levels, and then for a prolonged period. We want to now test it under this contract for its, essentially we are doing the dosing studies with this construct, this Catabody construct, in which we want to prove that brief treatments successfully remove the brain amyloid. Now remember that there are no drugs available right now for treatment of amyloid in the brain in Alzheimer's disease.
Well, that's of course not quite true now, right? We do have Aduhelm or aducanumab, as it's well known, and it looks like we're going to have another one, lecanemab, within the next few months. But these are more conventional binding antibodies, and their effect is not amazing. And the anticipation is that a Catabody would be much more effective. Let me take a short break, and then I can get your response, Dr. Paul.
Okay, very good.
Yeah.
So, Michael, I agree with your not quite true statement, but let me give you my take on where the field stands. I think there is an ameliorative effect of conventional antibodies in humans, as shown by the latest Biogen ASI antibody, the one that has not been approved. It is certainly a small effect, but a real effect seen after months of treatment with the antibody. And it is not really reversal of the the disease. It is slowing of the worsening of the disease. That's the latest, most promising unapproved antibody. The predecessor of that antibody, the one that was controversially approved by the FDA in the teeth of opposition from experts in the field, aducanumab, that one has a dubious effect seen in one trial, a very small effect, but it is, as I say, it wasn't seen in the second trial, and in fact, Medicare is not even covering the costs of that, so it's not being used. It's been essentially left
Not
to
not covering the full cost. Yeah.
And it is approved, but it is no, it's not being approved at all routinely. It's only if they are in a clinical trial, if it's a comparator, that antibody in a clinical trial, then it's covered. So essentially, Biogen is not hoping to make any profit out of it, and it's withering on the vine, so to say. But the second one is more promising, and we'll see what I think the approval will go through most likely. However, the effect is small and disease is not reversed. What is the problem then with the conventional antibodies? In my view, which is supported by the majority of the field, these ARIAs that are seen, these radiological abnormalities that are called side effects, are not really only side effects, they actually cancel the benefit of the amyloid removal effect of the antibody. The reason is these antibodies do not destroy the amyloid. They require phagocytes to take up the immune complex, which causes inflammatory effects in the brain, neuroinflammation, plus you get damage to blood vessels, and we are inducing cerebral amyloid angiopathy, which happens anyway in Alzheimer, but that is exacerbated by administration of the antibody. So it's not merely, it's wrong to look at it only as a side effect that is transient. The point I'm trying to make is that there's an actual cancellation of the beneficial effect of amyloid removal. Catabodies are, in our published work, we've shown that catabodies are free of this effect. Why? Because the antibodies digest the protein, the amyloid, very quickly, and the phagocytes remain unactivated. We see no signs of inflammation in the brains of the mice that we cured, so to say, in the laboratory, in our own experiments. So there is hope that the effect will be far more quick because it's a direct cleavage, direct breakage of the amyloid. We are not dependent phagocytes, so it'll be weeks of treatment at most, not months or years of treatment as with conventional antibodies. So that's one hope with catabodies, a mechanistic difference that catabodies enjoy. The second is the benefit of no inflammation, no cancellation of the beneficial effect due to the neurotoxic effects of inflammation. These are two major differentiating features that we think will bear out in studies that we have initiating with a large pharma company, and hopefully we can progress then to the clinical trials. And of course, we are keeping in science until the job is done. The predictions are only predictions. So in our work also, we have de-risked our project substantially, but it is a first-in-class product, and we'll see what happens. I'll leave it at that.
Yeah, I think that is a scientifically adequately cautious statement to make, but it's very promising. I mean, the Catabody technology is incredibly exciting for all the reasons you've outlined. It actually degrades it on site completely, so you're not trying to just mobilize the stuff out of the brain. You don't have to drag in a bunch of activated immune cells that can cause inflammation in the brain. You don't sort of remobilize the A beta into the blood vessels. you're just like getting rid of the A beta altogether and you're done. And you're doing it much more rapidly. So, as you say, we will see what happens in the event once you're, knock on wood, get to clinical trials. But right now, conceptually, it's an extremely exciting technology, and your animal results to date are really promising on that. Okay, so we've sort of gotten into this already now, but what's next?
Yeah, so there are, in my old age, I say it's a sort of an embarrassment of riches, choosing things a little tough with limited money that we have. These are first class drugs, so we need very strong basic science data and then translational data. What is next is to capitalize on the evidence, prove what we've just discussed, that these predictions are going to be correct with respect to car design by doing dosing studies showing that briefness, showing the benefits of brief treatment, non-inflammation with car design, ditto for Alzheimer's. And hopefully in collaboration with companies that are doing late stage work and have the expertise to take it further into human studies. Then we are moving forward with our toolbox approach to developing therapeutics and diagnostic agents. We have started a project on a chemokine that that is attractive because classical reversible binding conventional antibodies are already available. They are in the market, both for diagnosis and for therapy. And we think that working with a simpler target is going to be helpful for our company. Our goals here are perhaps for a startup company like mine, they are very lofty, and it'll be good to prove our points, our scientific principles, using a simpler target that we have started a project on. It is an attractive target for that reason, and our milestones are to prove against side by side with the classical reversible antibodies versus our Catabodies and IRABs, irreversible antibodies, that we can treat better, do better therapy, and do better diagnosis, ultra-sensitive direct diagnosis for early disease detection, owing to that infinite affinity binding that we discussed. So we have several, again, financing is a big deal. That's an overarching, it takes up a lot of my time to just convince people or to discuss people all these do's and don'ts and the pitfalls and the strong points of the technology. That is the big deal for us to convince peers and people with resources, companies with resources, to collaborate with us. And we think our toolbox we are optimizing our toolbox as the next step, high throughput procedures to shorten the time of delivery of these Catabodies and IRABs using new targets, as I mentioned, like this chemokine target, to within a couple of months or maybe 4 months at most, and not year-long basic science efforts. I think that's pretty much a roundup of the next steps for me.
Fantastic. Okay, well, as you say, there's financing required. There's doing the science is required. We're wishing that everyone goes forward as rapidly as possible and we get as quickly as possible to seeing some human rejuvenation. Good luck to you, Dr. Paul.
Thank you. It was a pleasure.